Assalamualaikum warahmatullahi wabarakatuh. My name is Aqilah Binti Ismail from Degree of Bachelor of Science in Biology, Faculty of Applied Sciences, UiTM Perlis. My final year project entitled Antibacterial Shower Gel from Clitoria ternatia Lin Leaf Extract, and my supervisor is Dr. Nurizawani Binti alias. Clitoria ternatia Lin from the Fabaceae family is known as butterfly pea or Asian pigeon wings that belong to a perennial twinning leguminous herb that is mostly found in tropical equatorial areas. It consists of about 60 species distributed widely around tropical regions, and few species can be found in temperate regions. Clitoria ternatia Lin possesses numerous biological activities which lead to various pharmacological benefits, especially for humans. According to previous research by Al-Snafi in 2016, Lijon et al. 2017, and Reka et al. 2018, Clitoria ternatia Lin comprises many phytochemical constituents such as tannins, phlebotanins, carbohydrates, saponins, triterpenoids, phenols, flavonoids, flavonol-glycosides, proteins, alkaloids, anthraquinones, anthocyanins, steroids, terpenoids, sterols, glycosides, and phytosterols. Some of the major chemical constituents found in Clitoria ternatia Lin extracts, such as tannins, phenols, flavonoids, flavonol-glycosides, alkaloids, anthocyanins, and sterols, serve a significant function that contributes to the potent antibacterial activity of this plant species. As leaf are the main focus of this study, Jamil and Pai in 2018 have proven that the leaf extract of Clitoria ternatia Lin possesses bactericidal action, causing it to be capable of performing powerful antibacterial activity against a few common bacteria such as Escherichia coli, Staphylococcus aureus, and Vibrio cholera. Due to its wide range of antibacterial activity, Clitoria ternatia became one of the most critical medicinal plants used in traditional treatments, since it can provide an advantage in increasing the effectiveness of the medical industry, as it has great potential in combating various emerging diseases and infections caused by antibiotic-resistant bacterial pathogens nowadays. In this study, an antibacterial agar diffusion assay is carried out to determine the antibacterial activity of Clitoria ternatia Lin leaf extract. Additionally, antibacterial shower gel provides greater antibacterial activity against various bacteria that either accidentally or inaccidentally settle down on human skin, which then infect the human body through unprotected and unhealthy skin. Due to its stronger antibacterial activity, Clitoria ternatia Lin leaf have been extracted for many years for use in cosmetics as well as body and skincare products such as shower gels. Therefore, the aims of this study are to evaluate the antibacterial potential of Clitoria ternatia Lin leaf and to develop the extract into an antibacterial shower gel. There are so many studies that have been conducted previously to determine the phytochemical constituents of Clitoria ternatia Lin root and flower extracts of this plant, while the leaf extract is given less attention for more in-depth study on its benefits and greatness. Therefore, the aims of this study are to determine the phytochemical constituents and to evaluate the antibacterial activities of Clitoria ternatia Lin leaf extract by using bacterial strains that live specifically on the human intact skin, such as Bacillus licheniformis from gram-positive strains and Escherichia coli from gram-negative strains. Additionally, the majority of Clitoria ternatia Lin root and flower extracts marketed are only concerned with their strong, sweet fragrance, which is calming, relaxing, comforting, and soothing, that consequently can cause skin irritation because the fragrance is the main cause of damage to the skin cells. Therefore, an antibacterial shower gel made up of clitoral tenatialin leaf extract can be developed and commercialized more extensively in the market since it produces less fragrance than roots and flowers. Some questions from this study are: what is the most suitable method that can be used for plant leaf extraction? What are the phytoconstituents that can be extracted out from the leaf root extract? How is to evaluate the antibacterial activity of leaf extract? What are the most suitable bacteria representing both gram-positive and gram-negative strains which can be used to determine the antibacterial activity of leaf extract on human intact skin? What is the concentration of leaf root extract of Clitoria tenatialin that can inhibit the bacterial growth after being mixed together with the liquid soap base? This study will provide new knowledge on the phytochemical constituents responsible for the potent antibacterial activity of Clitoria tenatia leaf extract. The study of the antibacterial properties of Clitoria tenatia leaf will prove the ability of this health beneficial plant to aid the medical field in battling against the emergence of various antibiotic-resistant bacterial pathogens. This study also will develop a shower gel which contains Clitoria tenatia leaf extract as it has good potential for commercialization as an antibacterial product. The objectives are to determine the phytochemical constituents of Clitoria tenatialin leaf extract by using phytochemical screening tests, to evaluate the antibacterial activities of Clitoria tenatialin leaf extract by using antibacterial agar diffusion assay, to produce an antibacterial shower gel of Clitoria tenatialin leaf extract. The leaf sample of Clitoria tenatialin was collected from regions all around Perlis. Then, the leaf extract was yielded by the solid extraction method and ethanol was selected as the solvent. Phytochemical screening tests were carried out to identify the presence of 13 chemical compounds, such as alkaloids, tannins, glycosides, steroids, saponins, flavonoids, phenolics, terpenoids, anthraquinones, leucoanthocyanins, coumarins, quinines, and also gum and mucilage in the leaf extract. Next, an antibacterial agar diffusion assay was performed to determine the antibacterial activity of the leaf extract against B. licheniformis and E. coli. However, the limitations in performing this study are the time-consuming procedure of plant extraction and also the lack of knowledge and skill to perform antibacterial agar diffusion assay. Muhammad Azzuddin and Rabita 2018 mentioned that this plant grows from a woody rootstock and could climb or trail. It is generally hairless on top and pubescent at the bottom. As stated by Muhammad Azzuddin and Rabita 2018, the flowers are axillary and can be single or two together in a racemopinated arrangement. They are enormous and beautiful with a vivid deep blue and white coloration. According to Anwar et al. 2021, this plant has a hairless or faintly pubescent pod, linear oblong with 6 to 13 cm long, flattened and mucronate at the apex. Muhammad Azzuddin and Rabita 2018 also stated that the plant's leaves are imparipinnate with 2 to 4 pairs of leaflets and a terminal leaflet that range in form from elliptical to elliptic oblong structure. It is naturally self-pollinated, but it also occurs because of cross-pollination following the discovery of genetic segregation. In a study by John Sushma et al. 2016, due to its potent biological activities, this plant species has been massively employed in Indian Ayurvedic, Siddha, and Unani traditional treatments. Gupta et al. 2015 in another study reported that this plant was utilized in urinary tract infections (UTI), burning sensation in the urinary tract, lack of urination, and frequent urination amongst the Tripura tribe of Bangladesh. Based on a study by Ramlan et al. 2021, the powder and decoction forms of Clitoria tenacia leaf are usually only used directly to treat various diseases such as diabetes, edema, mental disorder, goiter, vitiligo, a disease with pale white patches on the skin, snake poisoning, toothache, eye disease, fever, asthma, and jaundice, besides being used as a blue food coloring. In a study by Kavita and Premalakshmi 2013, the phytochemicals are chemical components which have a specific physiological function on the human body that helps in the therapeutic efficacy of plant species. Phytochemicals are not edible but have protective or disease preventive qualities. Phytochemicals have complementary and overlapping modes of action in the body which allow them to contribute to many vital biological activities such as antioxidant, antibacterial, antifungal properties, and many more. According to Kavita and Premalakshmi 2013, Clitoria tenacia leaf contain many phytochemicals. Nevertheless, phenols, flavonoids, alkaloids, and tannins are the major phytochemicals found to be present in the leaf of Clitoria tenacia lin. A previous study by Singh et al. 2018 reported that antibacterial activity is the action of killing bacteria, inhibiting their development or preventing them from reproducing. Antibacterial properties can be found in heat, substances like chlorine, and also antibiotic medications. Antibacterial agents can be categorized as either bactericidal, killing bacteria, or bacteriostatic, slowing the development of germs. Antibacterial agents are crucial in the battle against infectious illnesses. Due to the widespread use and abuse, bacterial resistance against antibacterial agents has emerged as a serious issue for today's pharmaceutical industry. Therefore, various medicinal plants have been discovered to be beneficial in the treatment of bacterial infections. Cyclotides and phenotine are two of the major components contributing to the great antibacterial activity in the leaf of Clitoria tenacia lin. According to Das et al. 2020 and August et al. 2019, cyclotides is a peptide that serves as potent antibacterial properties in Clitoria tenacia lin leaf to effectively kill the bacteria or inhibit the bacterial growth, mostly against gram-negative and some gram-positive bacteria. While phenotine, Camilla et al. 2009 and August et al. 2019 mentioned that it is the main protein component that possesses antifungal, antibacterial, and also insecticidal properties in Clitoria tenacia lin leaf. This plant species has various biological activities other than antibacterial activity. August et al. 2019 and John Sushma 2016 stated that the leaf have biological activities such as diuretic, nootropic, anti-asthmatic, anti-inflammatory, analgesic, antipyretic, antidiabetic, antilipidemic, anti-arthritic, antioxidant, vascular smooth muscle relaxant, hepatoprotective properties, and also wound healing properties. It also serves as antiemetic, antidipsetic, mind laxative, and cholagogue to treat diseases such as emesis, dyspepsia, and constipation, as well as working as an expectorant and soothe respiratory organ inflammation. Hence, it can cure colds, cough, and also asthma. Antibacterial shower gel provides better protection of human intact skin against various bacterial infections which are capable of threatening the human's health. The formulations is enhanced by the addition of Clitoria tenacia lin leaf extract, which possesses more potent antibacterial activity among other medicinal plants, as this plant is already widely known for its capability to kill various bacterial pathogens which can cause severe infection if they succeed to penetrate through the human skin barrier. According to Vogt et al. 2014, surfactants, preservative, acidity modifier, and pH regulator are four main chemicals commonly used to formulate a shower gel. Fresh leaves of Clitoria tennatia were collected from regions all around Perlis and were packed in a plastic bag. Then, the leaf samples of Clitoria tennatia were washed cleanly and air-dried and also shade-dried for a week. The dried leaves were ground into powder form by using an electrical blender. The dry and powdered leaf samples were weighed at 82 grams and later they were kept in an airtight sealed plastic bag. The leaf sample was extracted with a solvent of 90% ethanol by using the solid extractor apparatus for 24 hours. Then, the leaf extract was filtered by using Wetman No. 1 filter papers and was concentrated under reduced pressure at 60 plus minus 10 degrees Celsius in a vacuum by using a rotary evaporator for dryness. The leaf extract was stored in a refrigerator for further use. Preliminary qualitative phytochemical screening tests were carried out to determine the presence of 13 phytoconstituents in the leaf extract, such as alkaloids, tannins, glycosides, steroids, saponins, flavonoids, phenolics, terpenoids, anthraquinones, leucoanthocyanins, coumarins, quinines, and also gum and mucilage. An antibacterial agar-Dis diffusion assay was conducted to evaluate the antibacterial activity of leaf extract against B. licheniformis and E. coli by measuring the mean of inhibition zones of both gram-positive and gram-negative bacterial strains against three different concentrations of Clitoria tennatia leaf extract, which are 10 ppm, 20 ppm, and 30 ppm. The positive control used is gentamicin, whilst 5% of DMSO solvent was used as the negative control. The test was performed in triplicates for both bacteria. Antibacterial shower gel was prepared by using the liquid soap base which was purchased online. To prepare 100 ml of antibacterial shower gel, 1 ml of 30 ppm diluted leaf extract was added into 99 ml of liquid soap base in a beaker and the mixture was stirred and mixed well. Eventually, the shower gel was kept in a bottle. The changes in pH, color, and scent, as well as the gel texture durability of antibacterial shower gel was tested for a month of observation. The percentage yield obtained from 24 hours of the solid extraction process is 5.57%. Out of 13 phytochemical screening tests, 6 chemical compounds were found to be present in the leaf extract, which are alkaloids, tannins, glycosides, saponins, phenols, and also coumarins. Meanwhile, steroids, flavonoids, terpenoids, anthraquinones, leucoanthocyanins, quinines, and also gum and mucilage were tested negative in the study. Some phytochemicals found in the leaf extract are responsible for various critical biological activities. Alkaloids, tannins, and also phenols are responsible for antibacterial activity. Coumarins has antiviral activity. Glycosides possesses antidiarrheal activity, and saponins serve as antidiarrheal, anti-cancer, and also antihelminthic activities. Hamza et al. 2021 reported that the leaf extract showed concentration-dependent increases in the inhibition zones. For both bacteria, the highest concentration at 30 ppm showed the highest mean of inhibition zones, whilst the lowest mean of inhibition zones was at the lowest concentration of 10 ppm. At an intermediate concentration of 20 ppm, it exhibited an intermediate mean of inhibition zones. This proved that antibacterial activity was the highest at 30 ppm and the lowest antibacterial activity at 10 ppm. An intermediate antibacterial activity was shown at a concentration of 20 ppm. B. licheniformis of gram-positive exhibited higher inhibition zones mean than E. coli of gram-negative. According to Anand et al. 2011, Balasundaram et al. 2011, and also Hamza et al. 2021, it is due to the differences in structure of cell wall components where gram-positive bacteria lack of thick outer membrane layer of their cell wall, causing them to be more susceptible to antibiotics and medicinal plant extracts than gram-negative bacteria. Camilla et al. 2009 stated that B. liginiformis and E. coli exhibited mild or moderate inhibition against all three concentrations of diluted leaf extract. The pH of shower gel ranged from 5 to 6 after being tested with pH meter. The color and scent of the shower gel remained unchanged, and the gel texture durability was in good condition after a month of observation for both open and closed bottles. Both bacterial strains showed no inhibitory effects when tested on the shower gel. This might be due to the lower concentration of leaf extract in the shower gel base, which leads to slightly lower zones of inhibition formed for both bacteria. Hamza et al. 2021 mentioned that the higher the plant's extract concentration, the higher the antibacterial activity of a plant species. The antibacterial activity of shower gel showed no inhibition, since the values of inhibition zones for both bacteria were less than 12 mm zones of inhibition, as the leaf extract concentration of 30 ppm can be considered too low when mixed with the liquid soap base, causing the inability to exhibit potent antibacterial activity against both bacterial strains.